Hello everyone, I am Janisa, final year student of MSc Physics of Sir Said College at Taliparamba, affiliated to Kannur University. Today, I would like to present on the topic the biomolecular recognition of diosmin with BSA, a spectroscopic and computational study. As we all know, the interaction between a protein and a ligand are essential to all biological processes. So, for the better understanding of the recognition process, the molecular and atomic level information of the drug molecule is needed. And also, it's an important area of research in drug design and development. Here, we conducted a detailed examination of the interaction of protein BSA with diosmin by different spectroscopic and computational methods, including UV spectroscopy, photoluminescence, and molecular docking. Some facts related to BSA are given here. Bovine serum albumin or BSA, a protein derived from the cow, is one of the extensively studied plasma proteins in the field of protein drug interaction. Here are several properties of diosmin such as antioxidant, anti-tumor, anti-cancer, anti-inflammatory, anti-diabetes and anti-apoptotic properties. Figure 2 and 3 shows the chemical structure of BSA and diosmin respectively. Now, I would like to turn to the methods and techniques we have used to study the interaction. Firstly, UV visible absorption spectroscopy, which works on the principle of Beer-Lambert's law. We have done two sets of UV titration experiments. At first, we have kept the concentration of BSA constant as 20 micromolar while increasing the concentration of diosmin. Next, a reversible method keeping the diosmin constant as 30 micromolar by increasing concentration of BSA have done. The absorption spectra were measured and tabulated to determine the binding constant as shown. The binding constant obtained shows that there is a strong binding between diosmin and BSA, and it also helps to predict the conformational changes of protein during interaction between drug. Thus, the mode of binding and affinity of BSA with small molecules like diosmin can be well accessed from UV absorption spectroscopy. Secondly, we have adopted the photoluminescence spectroscopy, also known as fluorescence spectroscopy. In this technique, we can analyze fluorescence from a molecule based on its fluorescent properties. Here, we recorded the emission spectra at an excitation wavelength of 280 nanometer by keeping concentration of BSA constant as 30 micromolar. Stern-Volmer constant, quenching rate constant, number of binding sites and binding constant were tabulated from Stern-Volmer plots and logarithmic plots. And also, the fluorescence studies shows that the intensity of BSA decreases on addition of diosmin, which confirms the proper binding. Finally, I would like to move on to molecular modeling method, which we have adopted for computational study. In this work, the docking of diosmin with macromolecule 4F fibers were done through Autodock software. The binding energy of BSA ligand interaction is analyzed and the results showed that diosmin binds with BSA by favorable binding energy. The cluster analysis of conformation for lowest binding energy is also analyzed. Now, let me conclude that all these experimental and computational studies gives us a clear picture of biomolecular recognition of diosmin with BSA. Thus, we can affirm that diosmin binds with BSA and may be used as a lead molecule for drug designing. I am thankful to the Department of Collegiate Education, Government of Kerala for the Aspire Scholarship. Thank you.